Welcome to the installation on the immune system for humoral immune response. So this topic is going to be talking about the activation of the B cells, how we get them going, differentiation of the B cells, how we create the plasma cells, how we create the memory B cells, immunological memory, tagging off of those memory B cells, what do they do when they see that same antigen again? And the different types of humoral immunity that we can have. We can have active immunity, where we create the plasma cells, create the antibody ourselves. We can also have passive humoral immunity, where antibodies are provided to us, but are not going to then provide us with any memory. We won't have any memory cells. We're just going to have antibodies. They're passed on to us. And then the different types of antibodies. We'll discuss those. Okay, and then how we can use antibodies in science too. I'll talk a little bit about monoclonal antibodies towards the end. So, off and running, the humoral immune response, the B cell arm of the adaptive defenses. Activation and differentiation of the B cells. All right, we know that B cells are activated when they bind to the antigens, or more specifically, the antigenic determinants, the little pieces, parts that they can recognize. They're actually going to cross-link them. There'll be multiple B cell receptors on our B cell, and when it sees the proper antigenic determinant and gets a whole bunch bound to it, and if it's the first time being stimulated, a T helper cell will say, yes, you have found the right combination. Please go off and make some memory cells and plasma cells. And that's what's going to happen. All right, we're going to create plasma cells that are going to be kicking out antibody like crazy. And we're going to create a little handful of memory cells as well to live on forever in case you see that specific antigen again. So most clones are going to become the plasma cells. We're just going to have a handful of the memories, and the worker bees are those plasma cells. They're going to secrete antibodies very specific to that antigen. The antibodies are going to look the same as the B cell receptors do at a rate of 2,000 per second. All right, so they are spitting them out for about 4 or 5 days is how long they're going to create them, and then those plasma cells will die off. They're going to undergo apoptosis. Those antibodies continue to circulate through the whole body, through all the humoral fluids, so the blood, the lymph. They're going to be floating around everywhere and binding to any free antigen that they recognize. And remember, that flags and tags them. It marks them for destruction by cells of the innate system, macrophages, for example, any phagocytes, or adaptive mechanisms as well. A little bit more on the fate of our clones. The clones that don't become those plasma cells, they are very special. They become memory cells. So they aren't going to kick out a whole bunch of antibody and die. They have now been given a pass to live on forever. And these are the cells that are going to provide that immunological memory, that special component that the adaptive immune system has that the innate does not. So when these cells come across that antigen again, whether it's a week from now, a year from now, 10 years from now, they will mount an immediate response to future exposure of that antigen. They don't need mama T helper cell to tell them it's okay. They are going to kick off, and they're going to make a faster response, a stronger response, more plasma cells, more antibodies for a longer duration. They are really going to kick that antigen hard. And here's an image showing that process. All right, so this is primary response again. The first time these naive B cells are ever coming across the antigen that they have been built to recognize, they are going to bind. Now these two over here don't recognize, but this one does. And this is that cross-linking I was mentioning, where they'll have the 2 B cell receptors bound to that antigen. That is going to tell this guy, I should start proliferating. What they don't show here is that T helper cell saying, yes, go right ahead. But that does happen. And then they start to clone out and proliferate. They're going to create lots and lots of twins. Some of those twins, those clones, again, are going to become plasma cells, the effector B cells, kicking out all that antibody. And a small subset will become those memory B cells. And these will respond if we see that antigen ever again. So let's look at this on a curve at the antibody production. This is that primary immune response we just looked at. Cells are going to proliferate. That usually happens for about 3-6 days. They're going to create peak levels of plasma antibody around 10 days, and then antibody levels are going to decline because the plasma cells are going to have died off, and antibodies made of protein, it's going to get beat up and degrade, so that will disappear over time. That's your primary immune response, a nice little blub. Now, if those memory cells get hit, secondary immune response, harder, faster, stronger. And here it is, that secondary immune response. Re-exposure of those memory B cells to the same antigen is going to give a much faster response. It's going to be more prolonged over time, lots and lots of antibodies being kicked out. Those memory cells can respond within hours, not days. We're going to peak our antibody levels in 2 to 3 days at much higher levels than our primary response. Look at that. And it can remain very high. Those plasma cells actually live a little longer in the secondary response than the primary first response. So you can have pumping out antibody for weeks to months when you see that antigen a second time, a third time, a fourth time. It's always going to look like this, stronger, okay, more efficient than those primary responses. And here's another primary response to a different antigen, a purple one apparently, instead of a blue one. So you've already seen this primary response, and we're going to drop that memory cell down so that when we see that antigen again, next month, next year, 10 years from now, we are going to have a faster, more prolific response. Plasma cells, lots of antibodies, and yes, another subset of memory cells. And I don't know why they showed this here, that you have 1 memory cell for the primary and 2 for the secondary. You're going to have a cluster. You're going to have multiple each time. But I thought that was kind of cute. Primary and secondary response taking care of ya. I mentioned different types of humoral immunity, active and passive. Active is going to be when you actively participate, your B cells respond to antigen. And there's 2 different branches of this. When your B cells see an antigen, bind to that antigenic determinant, it's going to happen just like you saw previously. We're going to have naturally acquired, or we're going to have artificially acquired. So for naturally acquired, you're actually getting exposed to bacteria, to virus, to parasites. It's actually naturally occurring, and your B cells are responding. Artificially acquired active humoral immunity is awesome because this is where we can use vaccines. This is one of the best things about those B cells and T cells, is those memory cells you can create. So we can actually use broken down, dead bits and pieces of pathogens to create that primary response to something that can't hurt us. Here's measles, mumps, and rubella broken up into bits and pieces, so that when we have an actual exposure to one of those very destructive diseases, like measles, mumps, and rubella, we'll have already had our primary response. So we are going to kick that virus's butt, measles, mumps, or rubella, harder and faster, and probably be able to survive those infections because the vaccine created that primary response for us. So most vaccines are going to be dead pathogens or attenuated pathogens where they've been modified just a little bit that they can't be infectious to us, but still expose all those antigenic determinants to our B cells. Right? It spares us the symptoms of the primary response while still creating it. And then we have the memory cells to help us out when we actually do become exposed to anything like measles, mumps, rubella, polio, smallpox. You can see this is from the 1900s, the numbers of cases of these different disorders. And now, obviously, having that primary response already taken care of has saved a lot of individuals and saved us from being infected from a lot of sometimes deadly diseases. Tetanus as well. So if we don't have active humoral immunity, we have the other branch, the passive humoral immunity. This is where our B cells aren't doing the work, but we are passively receiving antibody from somebody else or from an injection from the doctor that was created in a lab somewhere. So these are going to be ready-made antibodies, okay, and they're going to be introduced to your body So none of your B cells are being challenged. We don't have a primary or a secondary response going on here. There's going to be no memory cells created. So the protection is going to end when those antibodies break down and degrade. And when might we want to use this? Say we got bit by a snake. We have pre-made serum, anti-venom toxin serum, that we can shoot up a whole bunch of antibodies straight to us to block up that toxin, not let us do any destruction. Tetanus as well. We've got tetanus toxin, anti-tetanus toxin serum that we can use. Mom to baby, antibodies can cross over to the fetus through the placenta. We can also provide antibody protection to the babies through breast milk as well. So these are all going to be short-term, but quick protection, not involving your B cells, but mom's B cells in this case, for making that antibody, or we've created some antibodies in a laboratory as well. And I'll talk about those at the very end with those monoclonal antibodies. So a little bit more on the passive humoral. Those two types are, again, naturally acquired, okay, getting from mom to fetus or milk to infant. And then we have those artificially acquired, where we're having an injection, all right, for tick antivenom, snake antivenom, yes, even jellyfish antivenom. All right, immediate protection. Don't have to wait for your immune response to rev up to these different venoms. We can take care of it. But again, these antibodies are going to degrade in your body, and you're not going to have that primary response. You're not going to have those memory cells created. This is a great figure that sums up everything we've just discussed. For the humoral immunity, you can have the active arm, which is where the B cells are actually doing the work, your B cells, either naturally because you came in contact with the pathogen, or artificially because you went to the doctor and got yourself a vaccine, flu vaccine every year, because flu changes so much in its antigenic determinants, whereas some do not change very often, like measles, mumps. That's why you can usually do a one and done on the vaccine, or maybe get a booster. The other arm is the passive, where you're just receiving antibody, right? Naturally from mom to fetus through the placenta, or from mom to baby through the milk. Passive you can also get artificially, right? Injections of antibodies that would be created in labs. So those are your actives and your passives. Let's talk a little bit more about antibodies themselves. Antibodies, these little guys here, are immunoglobulins. Globulins should automatically make you think proteins, and these are part of the immune system. So they are immune proteins we use for the immune system. If we spun down some whole blood, where do you think these immunoglobulins would hang out? Yes, if you said here in the serum, you are absolutely correct. That's where those antibodies would be located. Okay, so they are proteins secreted by your plasma cells, the activated B cells, and they are capable of binding specifically with one antigenic determinant that B cell was trained to find. What I haven't shared yet is that all antibodies don't look like this Y. All right, there are actually 5 different types of antibodies. There's IgM, which is the pentamer. There's IgA, which is a dimer, so you have 2 structures of the antibody. And then there are 3 types of monomer antibodies that look like this. It's D, E, and G. So how we remembered flat pig for some hormones and never let monkeys eat bananas for antibodies, you can look at MAD egg. M, the pentamer, A, your dimer, and DEG, the 3 monomers. MAD egg. And they'll have different jobs. They'll occur at different times of infection and different locations in your body. Let's look at the structure real quick, and then we'll look at where they're located in your body. Basic structure. All right, don't look at this and freak out. All right, I'm not going to expect you to memorize this and label where the light chain is and the heavy chain is. I just want to give you an idea of the structure. Right, we know this is protein, and it's actually made of 4, all right, there, looping polypeptide chains, and they're held together 1, 2, 3, and 4 by disulfide bonds, which are these yellow bonds. You have 2 heavy chains, and they are identical when they are produced. There's your 2 heavies, longer, bigger, heavier. and 2 identical light chains, these lavenders here on the side that will attach and make that Y formation. Now the variable region is the most important, and the variable region is the ends of these 2 light and heavy chains over here. That's where we get all the different antigenic determinants we can see because there's such variation at the ends of these. Those are the antigen binding sites. This end here is the flag for your phagocytes or for your complement to be activated, but these 2 ends here, the variable ends, that is where we have all of that diversity for our B cells and our antibodies. Let's talk very briefly about the 5 different types, where they are, when they're created. IgM, which is the pentamer, it's the largest out of all of them. That is actually the first antibody to be released in a primary immune response. All right, so all the images they showed before, they just made it simple and showed you the monomer, but it's actually IgM, the pentamer, that's thrown out in gobs at the very beginning of a primary immune response. And that makes sense. You know, you want to send out as much as you can. So here you'll have 10 different binding sites to throw out there and grab up as much of the infection as possible very quick. So it's going to be a very potent agglutinating agent, grabbing all that antigen. Okay, it's also very good at activating complement. IgA is the dimer. This one is going to be located in areas where there's mucus and other secretions. So IgA is going to help prevent entry of pathogens to those mucosal lined areas. The 3 monomers. IgD. IgD is not a free-floating antibody. It is actually the B cell receptor. All right, so this is the monomer attached to the surface of the B cell, and it would attach right there. IgG. This is the big mama. Okay, it is a monomer, but it accounts for 75 to 85% of the antibodies in your plasma at any given time. I mentioned in the early primary response, you get those IgMs, these guys right here, and after a little bit, those plasma cells switch over from producing the IgM to producing this IgG. So it is produced in the late primary and all future responses. So your secondary, your third, your fourth, your 20th response will always be IgG out of those memory cells. Right. IgG is also one that can cross that placental barrier. So when mom is with fetus and sees something for a second time, a third time, the memory cells produce that IgG. That is then transferred over to help protect the fetus as well. Last but not least, mad egg, IgE. All right, this is the least populous of the antibodies. It is a monomer, and it's active in some allergies, some parasitic infections, which means mast cells and basophils are going to be releasing their histamine caused by the IgE, but this is the least populous. All right. What I want to emphasize with all these antibodies is that it doesn't matter what class it is. So if it's IgG, if it's IgM in that primary response, they are going to respond to the same antigenic determinant. The variable region up here for IgM and IgG in the primary response are going to be the same. They're just changing the structure of the base for the heavy chain to either be a 5 pentamer or down to the single. All right, but we're going after that same antigen. I included tables from the book. This goes into a lot more detail for those of you that want a little more knowledge on the B cells and their antibodies. But again, what I'm more concerned about is what I just previously showed you. All right, primary response for the IgMs, IgAs are going to be in areas where we do secretions, so saliva, sweat, intestinal juice, milk, in the mucosal membranes. That's where we're going to have these guys, the IgAs. The monomers D, E, and G. Again, the Ds are just the B cell receptors, so they're found on the surface. The IgGs are the most populous. They're going to come late in the primary response and for all secondary responses. And IgE is the least populous. This is going to be tying in with releasing some histamine from basophils and mast cells, so this is going to be part of inflammation and allergic reactions. On to the antibody targets and the functions of antibody. I've mentioned some of these in previous lectures. We know that antibody, here's an IgM here, definitely causes agglutination when you did your blood labs. Okay, it's going to clump up those cells. I've already talked about complement, creating the MAC attack, and how antibodies can activate complement. Don't know if I've mentioned yet precipitation, where antibodies will attach to soluble components, say the toxins from that jellyfish. And if antibodies also coat things like viral particles so they can't enter the cell, bacteria as well can be coated. That process is called neutralization. So antibodies, again, they inactivate antigens, they flag them to be destroyed, but they do not destroy the antigens themselves. They are a defensive mechanism, and we use them in these 4 different ways. So let's run through. We'll start with neutralization. Neutralization, and I did little cutouts up here. So neutralization is simplest defense mechanism. If you coat that virus, if you coat that bacteria, it can't get to you. And this is very prevalent here. I can show you with SARS virus. If it is not coated, it can bind to receptors on your cell surface and enter your cell. All right, but if we have it coated with antibody all the way around, neutralizing it, it cannot get to those receptors on the human cell surface that is adapted for to be drawn into the cell, and we have protected our cells from SARS viral infection. That's neutralization. Agglutination. Agglutination you saw when you did the mismatched red blood cells. Okay, the antibodies bind to the same antigenic determinants on more than one cell. Because remember, we have that Y, so we can bind to 2 cells, hold them together. Another antibody can bind to another cell and hold it together, and it's going to create this clumping. Okay, that's agglutination. Precipitation is very similar to agglutination. You can see how we're pulling everything together, multiple bindings, causing this clumping, but it's not with cells. It's with soluble antigens, is what precipitation is. All right, so it's agglutination of small molecules. And then this will be subject to phagocytosis, just like we can have with the agglutination of the whole cells as well, such as bacteria and the red blood cells. So precipitation, again, extremely similar, pretty much the same thing, but with small molecules instead of whole cells. Last but certainly not least is activating that complement, okay, creating that MAC attack with those 20-some proteins that we have with complement, creating those pores. Mismatched red blood cells, bacteria are all subject to this complement activation and is our main antibody defense against cellular antigens right there. All right, cell lysis being the result. In this figure, you can see all 4 that we just spoke about: neutralization, agglutination, precipitation, and complement activation. So let's run through this real quick. Antibodies find the antigen of interest, and we then have antigen-antibody complex. That's what all 4 of these are. If we have that antigen-antibody complex, such as over here, to activate complement, we're going to have cell lysis. If you recall, there are a few of the complement proteins that also enhance inflammation and also enhance phagocytosis, okay, because they bind to the surface as well, just like antibody does. If we go down the other arm with this antigen-antibody complex, we can inactivate the antigens by neutralization, surrounding them and wrapping them, agglutination, binding to cell-bound antigens and clumping up cells, or binding to soluble antigens and clumping them up in precipitation. All 3 of these, if we follow the arrow, lead to enhanced phagocytosis. So this is how the antibodies help us remove these naughties. Block them up, let them get eaten, help to activate complement to lyse the cells, and also help to enhance phagocytosis and inflammation. Go antibodies. All right, when I spoke earlier about the passive immunity, let me see if I can roll back to this slide here for you real quick. Bam, right there. The passive humoral immunities, where we had artificially acquired antibodies by a serum injection to protect ourselves from ticks and, jellyfish venom, things like that. We don't draw these out of people in little small batches. We actually make them in quite large batches in laboratories, and we will do that by a special process to make all those perfect antibodies against that one antigen using monoclonal antibodies. So one clone. These are commercially prepared for that single antigenic determinant. So what do? We're going to inject an animal with that antigen of interest. All right, that is going to then activate those B cells. We're going to isolate those antibody-forming cells and divide them up into individual cells. So one cell in one petri dish, one cell in another, and we actually fuse that cell with a cancerous cell, a tumor cell. Once we fuse those together, it's called a hybridoma. And a hybridoma is, again, a fusion of a tumor cell and a B cell. So now we've created an immortal B cell that's just going to do nothing but divide and pump out antibodies against that specific antigen. So then we can take the media that we leave these cells in to feed and proliferate on and drain off the media, give it some fresh so that it can keep producing antibodies, spin those antibodies down, and create those nice little vials that the ER will have in case you need it. Not only do we use hybridomas to create serum, but we can also use them in research. If there's antibodies that I want to mark a specific cell, tag it with a fluorescent tag, and then look at it under a fluorescent microscope, I can do that. Anything that we can get as antigen and inject, we can create these cells, create these hybridomas. It's a pretty common process, and create these monoclonal antibodies. We find some that might fight cancer. Start working on those for clinical testing. I worked a lot with monoclonal antibodies tagged with fluorescence, tagged with metals, and then we could actually use magnets to draw out what they bind to. It's a way to select out components in columns. Wide uses for this. Love, love, love. Summary for the antibody actions. Again, we are looking at antigen-antibody complexes. They do not destroy the antigens, but they prepare them for destruction using all those wonderful innate defenses like complement, like phagocytosis and inflammation. They draw that out of us, but do not kill themselves. They are placeholders. Antibodies do not invade tissue. That's why they're considered humoral. They're out in the fluids unless there's a lesion present. If there's some damage to the cell that they could get in, then yes. But for the most part, that's why this is called the humoral side of the immune system with the B cells, because it attacks invaders outside the cells. Guess what the next topic is going to be? Future lecture, right? We're going to talk about the cellular immunity arm, those lovely T cells that take care of once virus actually did get past all that antibody and it wasn't all blocked up, it did get in the cell, or cancerous cells, or there's a parasite that got in the cells, whatever. That's when we're going to kick off the T cells, and that's the cellular immunity side of adaptive immunity. That will be the next lecture. Talk to you later.